Welcome to Beyond Lab Walls, a podcast from the Salk Institute. Join hosts Isabella Davis and Nicole Milnarek on a journey behind the scenes of the renowned Research Institute in San Diego, California. We're taking you inside the lab to hear the latest discoveries in cutting-edge neuroscience, plant biology, cancer, aging, and more. Explore the fascinating world of science while listening to the stories of the brilliant minds behind it. Here at Salk, we're unlocking the secrets of life itself and sharing them beyond lab walls.
Hi everyone, and welcome to Beyond Lab Walls. I'm Isabella, and today I'm really excited to sit down with Professor Tony Hunter. Tony is a cancer biologist, but that happened kind of accidentally. His real interest has always been in the basics, exploring the intricate programming and regulation of the proteins and molecules that rule our cellular world. His dedication to the basics has led to some incredible discoveries about cancer biology, which have been used to develop therapeutics that help real patients fight cancer every day. This year is Tony's 50th year at the Salk Institute, so we have quite a legacy to cover today. I'm going to go ahead and jump in at the very beginning. Hi, Tony, welcome. Where'd you grow up, and were you always interested in science?
So I grew up in Kent, in the southeast corner of England, near Canterbury. I wouldn't say I was always interested in science. My dad was an MD, he was a surgeon at a local hospital. My grandfather was a dentist, so there was sort of medical science in the family, but I wasn't really deeply passionate about science. And it wasn't really until I was sent away to a boarding school in Essex at the age of 13 where I sort of developed an interest in science. In fact, there's a funny story that when I got there, I was put in the lowest possible grade. Within 2 weeks, it was decided I needed to move up a grade. And so the headmaster called my father and said, Tony's got to be moved a grade. Should he specialize in science or in the humanities? And the decision was made, without my consent, that I should specialize in the science classes. And so from the age of 13, really, my career was set. When I graduated from high school, or public school in that case, and went up to Cambridge for my bachelor's degree, I focused on all natural sciences, as they were called, and particularly specializing in biochemistry in my final year.
So I know that Oxford and Cambridge have a unique teaching style. In addition to normal lectures, you meet with an advisor to talk through what you know and what you've learned on a weekly or bi-weekly basis in very small groups. And on top of that, of course, you sit exams and all of that like a normal student, but science homework can be written instead of in pop quiz form or in worksheet form. I'm curious, in retrospect, do you think that intimate style and the emphasis on written and spoken explanations of your science shaped the way that you do science and your desire to come to Salk, which is another place that similarly emphasizes relationships and collaboration?
Well, I'm sure it had a big effect on my development as a scientist. Yes, in Cambridge they're called supervisions and not tutorials. And so for each subject, you have a supervisor who you meet once a week, I think it was, maybe less, and talk about the lectures you'd just been listening to. And supervisor could fill in some details and explain some things you didn't understand. So I had some very influential supervisors who went on to become very eminent scientists. And then in turn, when I graduated with my bachelor's degree in 1965 and became a graduate student, I then took on the role as a supervisor. And some of the people I supervised have gone on to be very influential. In fact, one of them went on to become the director of the famous Laboratory of Molecular Biology in Cambridge, where Francis Crick and Sidney Brenner, Aaron Klug were.
Yeah, there's Salk connection there.
So you were studying biochemistry at Cambridge, you worked your way up to supervising. When did you pivot to cancer biology?
Well, that took a long time, actually. I was really trained as a pseudo-molecular biologist. So in the mid-60s, when I started my graduate work, the genetic code had not yet been fully worked out. I mean, we knew DNA encoded proteins, and we knew a little bit about messenger RNA, but the genetic code was still being worked out. And we couldn't do any of the things we take for granted now, like sequencing DNA or sequencing proteins, etc. So I was interested in how proteins are made in cells, and that was what my graduate thesis work was on. And then when I got my PhD in 1969, I remained in Cambridge as a college fellow. But in 1969, right after I got my PhD, I married a fellow graduate student, Pippa Marek, and she was training as an immunologist a couple of years behind me, and she decided she wanted to do a postdoc in San Diego at UCSD. And so I had to find a place to work in La Jolla for a couple of years while she did her postdoc. Luckily, her advisor knew Walter Eckhardt, and I met up with Walter at a meeting at the Royal Society in London, and I went up to him and said, would you take me on as a postdoc? And he said yes. I hadn't realized really at the time that he hadn't had any postdocs. He was just a new assistant professor. He wasn't called an assistant professor. They were members, I think they were called. So I became, actually in the end, his second postdoc, and we arrived here in October of 1971. So this huge social change from being in rather formal and proper Cambridge to the very relaxed California lifestyle. But Walter's lab was working on a DNA tumor virus, polyomavirus, a small circular genome virus, and trying to use it as a model to understand potentially the basis of human cancer. And so I started working on polyomavirus, thinking that it would be temporary and I would go back to Cambridge, finish my fellowship, and get a job working on protein synthesis, probably. And then I did go back. Pippa and I split up. She got a job in the US with another partner, and I went back to Cambridge to look for a job, which I applied to a couple of institutions, and neither of them wanted me. So I was working in the Department of Biochemistry in Cambridge, which is where I got my PhD, and was working as a research fellow. So I applied to them because they had an open position, junior position. And I also applied to the Imperial Cancer Research Fund, which was a cancer research institution in London, in Lincoln's Inn Fields. And Renato Dulbecco, who had been one of the founding fellows at the Institute, he was the first fellow to arrive and start work in the temporary buildings, and then moved into the third floor of the North Building in 1966 when the building opened. And he was married to Maureen Dulbecco, with whom he had a child, Fiona. And I think they decided that they would like to bring up their child in the UK. And so in 1972, Renato left the Institute, became the vice director of Imperial Cancer Research. And so I wrote to him when I got back in 1973 and said, "Dear Renato, do you have a position for me?" And I got this letter back that said, "Dear Tony, thank you for your inquiry about a position. I regret to say that we already have enough molecular biologists." Of course, they didn't really have any molecular biologists, but that was his excuse. But his leaving the Institute created half a floor of empty space because every fellow had 8,000 square feet, and there weren't any junior faculty at that time to work in the general area of tumor virology and cancer research. And before I went back to the UK, Walter had offered me a position as one of these junior faculty members and assistant professor. And I had turned him down because I thought I would go back to the UK and get a job there. But when that didn't materialize, I called up Walter, or I wrote to him, I forget now what I did, and asked him whether the job was still open. And he said yes. much to my relief. So I came back in 1975. I had to wait for a while. I think I got the job offer in May of 74, but I couldn't come back until I got a green card, and my green card was delayed because there had to be a local police report as part of my dossier to, I guess, ensure that I hadn't been arrested or done anything illegal. And so it took about 6 months before I actually left the UK. And during that time, we managed to burn the lab down. There was no sprinkler system and no one else in the building.
How did the fire start?
The official verdict is it was an ether wash bottle in a refrigerator that had been sparked by the internal thermostat. My version is that I think it was a water bath over one of the cup sinks in the lab, because we used to use ether for extracting TCA and then pour it down the sink. And so there was a layer of ether on the water remaining in the drain. And when I went into the lab around, 10:30 that night, I could smell ether in the air because it was an unusually warm June evening. And either way, it burned, and it burned for several hours.
And Salk didn't see that as a red flag that you might burn down one of their labs?
Well, I guess it could have been, but actually the offer was made before we burned down the lab.
So outside of having no other choice, since your Cambridge lab burnt to a crisp, were there other factors that pulled you back to Salk?
Well, one of the motivations for coming back was that we had started river rafting. I was a postdoc. Bill Cogan, a graduate student in the lab, had done a rafting trip in 1971 with some friends, and he decided this was such fun. They rafted the Colorado River through the Grand Canyon, that he would organize a trip for the summer of 1972, and he asked whether we'd like to come, and we didn't really know what we were letting ourselves in for, but we agreed. And so we met up with this rather chaotic trip. We had all sorts of equipment problems, etc. For the next 2 weeks, we rafted down the river, going through all these wild rapids and flipping some boats, etc. And the day I hiked down was the last day I shaved. So that was July the 13th, 1972. So I have not shaved once since then.
Wow, to this day.
The 70s, and people were growing their hair long. And actually, there's another funny story about long hair. So my hair, I'd actually grown it a little bit in Cambridge, but it was quite long. We used to go camping in the Baja Peninsula as a postdoc. The Mexican border agents were trying to, prevent drugs, particularly marijuana at the time, from coming into the US. And so they would check people going down to see if that's what they were potentially planning to do. And so knowing that, I'd bought a wig to tuck my hair under, so I looked like I was relatively short hair, even though I had a beard. And so we were driving through Tecate, which is a much smaller border crossing, and the agent there, I drove up and he looked at me and he reached over and whipped the wig off my head. And so we couldn't cross there, so we turned around, and then we were a group of 3 or 4 cars, and I was put in the back of another car with my hair pinned up with a couple of kids in it, and we got through in Mexicali instead. And so that was suddenly, all of this was clearly, influential in my decision to come back.
From Cambridge gowns and traditions to wild rapids and having a wig snatched off your head by a border agent. Wild transition. It seems like you adapted to the California lifestyle really quickly. At this point in your life, do you still feel British?
I have, yes. I left the UK for good when I was 31, I guess. Is that right? Yeah, 31. And so obviously, yes, I spent more of my life here than... I still think of myself as British, I think. Although I'm a US citizen, I'm a dual citizen.
So you have these adventures, you make it back to San Diego, you set up your lab. What are you working on?
I came back into the same lab space where I had been as a postdoc, right next door to Walter. We continued to collaborate. Franca and I published about 7 or 8 papers as Hunter and Franca, or Franca and Hunter, based on the work we'd done as postdocs. And Walter's name wasn't on the papers, and I just think we acknowledged him for providing support. You know, it had been a great collaboration, and I'd learned everything from him about polyomavirus. And so when I came back, it was natural. I would have to switch back to working on polyomavirus, and there was funding available. In fact, while I was back in the UK waiting for my green card, I had written my first grant application to study DNA replication that I had been working on it as a postdoc, and got that funding. And so I sort of obliged, I think, to do cancer research. In the interims, in the year and a half that I was back in England, quite a lot of progress had been made in understanding the polyomavirus genome. And so one could begin to decode the proteins that were made by the virus. And the key, obviously, to understanding how polyomavirus causes tumors is to know what proteins it makes. The virus has 2 halves, really. One half makes the coat proteins that package the DNA, and they're clearly unlikely to be involved in how the virus transforms cells into cancer cells. And the other half of the virus we call the early region, because it's the part that's transcribed into RNA and translated, that when the virus enters the cell, we could deduce 2 lines of evidence. One was we had antibodies that pull down proteins from the infected cells that we call tumor antigens or T antigens, and there were 3 of them, and we could deduce from the DNA sequence how they were made. So that was occupied the first 2 or 3 years of what I was doing when I came back and set up a lab, was to try and figure out how these proteins are made, which of them are most important for making a cancer cell.
You really were digging into the cancer biology then at that point.
I continued a little bit of work on tobacco mosaic virus. That was the collaboration we set up in Cambridge after the fire. There was a group working on the structure of tobacco mosaic virus. And the puzzle was how this RNA, it's an RNA virus, it packages a messenger RNA, so the RNA is translatable. When you took the RNA and put it in one of these cell-free translation systems, it does not make the coat protein. So this was a big puzzle. Mm-hmm. We knew the RNA encoded the coat protein based on some mutation studies, but it didn't make it. And Tim Hunt and I, Tim was a fellow graduate student in Cambridge. He went on to win the Nobel Prize for his work on a cell cycle. Figured out that the virus must somehow make a smaller RNA that makes the coat protein, and that's what we discovered.
It's incredible how technique has advanced so much in such a short amount of time, especially when you're looking at RNA and DNA, where sequencing a genome was not too long ago this giant puzzle that took enormous effort.
Well, the human genome, was sequenced only 25 years ago now. It took just a huge amount of effort and billions of dollars, and now you can do it in a few hours on one of these machines.
So you're learning more about how tumors form, and along comes this really iconic discovery of yours. I want to ask you to tell that story, but I want to also give our listeners a little context first. So the cellular world is run by proteins. Proteins determine everything from cell identity to specific cellular functions, and one way they do this is by sending signals. They can send signals within one cell, or they can send signals to other nearby cells to warn them of important news. But obviously, they can't always be sending messages. We want our proteins not crying wolf. Instead, cells have special ways of turning proteins on and off and regulating their activity. One of those ways is through the use of tyrosine kinases. Tyrosine kinases modify proteins by phosphorylating them, which means they attach phosphate molecules to them to regulate their activity. Tyrosine kinases specifically control proteins that help our cells grow and divide, 2 things we don't want cancer cells to be able to do. Figuring out how tyrosine kinases work could help scientists control them in cancer or other diseases where we want cells to stop dividing. And that's what Tony set out to do. I will let you tell the story now.
Once we'd identified that one of the 3 polyomavirus encoded tumor antigens middle T was the main factor through which the virus switches the cell into a tumor cell. Basically, it drives the cell into the cell cycle so it can replicate, and that then continues on to become a tumor. We wanted to know how it did it, and we really didn't have any good ideas, but luckily Ray Erickson's group in Denver had shown that the transforming protein of sarcoma virus has an associated protein kinase activity, and their rationale was if you have a temperature-sensitive mutant virus that only transforms cells at a low temperature, 36 degrees centigrade, and then shift the cells to 41 degrees, the cells, they go from a round transformed phenotype to a flat normal phenotype. That happens extremely rapidly within 30 minutes. If you now shift those cells back again to the permissive temperature, within 30 minutes or so, they become transformed again. So they deduced that whatever was happening probably didn't need new RNA and protein synthesis. It probably was happening on proteins already in the cell. We knew that phosphorylation was a way of changing protein activity. And so they tested the idea that maybe what was happening was that the temperature shifts using antibodies to the Salk protein, they showed it had a kinase activity. And of course, this was really dramatic and exciting. We immediately thought we should test whether polyomavirus proteins have this kinase activity. Much to my delight, we found the middle T antigen protein became phosphorylated in an in vitro kinase assay where we pulled down the proteins and then incubated them with gamma P32 labeled ATP, and that led to the labeling of the middle T protein. Wasn't seen with mutant polyomaviruses that couldn't transform cells. So that was an observation that was made by two other groups. And we all attended the Cold Spring Harbor Symposium in the summer of 1979 and agreed to submit our papers to Cell, which was the up-and-coming journal, to publish back to back to back. But the day after I had submitted the paper, I realized that one of the reviewers would probably ask what amino acid was getting phosphorylated, and it turned out that that was tyrosine, which I had learned about in my biochemistry courses in Cambridge as the third hydroxy amino acid. And so eventually, by making some phosphotyrosine, we could establish that the middle T was phosphorylated on tyrosine. But then it was important that we were also working on sarcoma virus, because Erickson's group had reported that the Salk protein was a threonine kinase. And so one day I was running the Salk protein as a control, and much to my amazement, it turned out that Salk was also phosphorylating tyrosine, and that Erickson had been misled by the fact that phosphotyrosine and phosphothreonine co-migrate at pH 1.9. And when I had done the original experiment, I had been too lazy to make up a fresh pH 1.9 buffer, and I did. Its pH had dropped 0.2 of a pH unit, and that allowed separation of phosphotyrosine and phosphothreonine. So it's the story in a nutshell.
It's so incredible the number of scientists and experiments it takes to come to a discovery like this. And today there are cancer drugs that target tyrosine phosphorylation. One of those is Gleevec, which is particularly effective in treating chronic myeloid leukemia, but is also effective against other cancer types. How does basic research turn into a therapeutic?
Well, that took a long time. Yeah. I mean, the discovery that two viruses apparently were using tyrosine phosphorylation to drive cancer was a start, but that didn't mean to say this was happening in human cancer, and that took a little while longer before people identified human cancer genes, and particularly, I would say, the BCR-ABL fusion gene that drives chronic myeloid leukemia was really the first strong evidence that tyrosine phosphorylation was driving a human cancer, particularly a leukemia. But then through various strategies, people found other fusion proteins that involved a tyrosine kinase gene. I mean, within about a year, we already knew there must be at least 4 human tyrosine kinase genes, because different retroviruses had captured different tyrosine kinase genes. Then it became possible to do cloning using PCR-based strategies, and that led to a whole new set of tyrosine kinases So more and more tyrosine kinases were being identified. Evidence that some of these were transforming tyrosine kinases began to accumulate. And so within 10 years, by 1990, we knew there were about 50 human tyrosine kinases, and about half of them have been implicated in cancer. And so that led to the interest in developing potential small molecule or antibody inhibitors, because several of the tyrosine kinases that were implicated in cancer were receptor-like kinases. So they were proteins that spanned the plasma membrane with an extracellular protein and an intracellular kinase part. And normally they're regulated by binding a small protein ligand like epidermal growth factor. But by being fused to another protein, they become activated through forming pairs or dimers of proteins. That's just how the ligands work. And so one can make antibodies against the external part of these proteins. So there were two real approaches. In fact, the first tyrosine kinase inhibitor drug to be approved was Herceptin, which is a monoclonal antibody that blocks the activity of a receptor tyrosine kinase called HER2. So that was in 1998, I think that was approved. And then small molecule approach, both in academia, but more importantly in industry at Ciba Geigy, that then became Novartis, led ultimately to the development of Gleevec. So it took, that was 20-something years. Now things are greatly accelerated. Then one had to develop all the technology and the libraries and things to develop the small molecule. Now there are something over 80 tyrosine kinase inhibitors that are approved. Many of them are targeting the same kinase, but they're different because each company has to have a unique chemical entity to get it patented and approved.
How specific are these drugs to certain cancer types?
Well, each drug is targeted towards one specific tyrosine kinase. And so any cancer that has a driver kinase of that type can be targeted by that small molecule drug. So it started with drugs that target leukemias, but now most solid tumors will have a potential TKI that can be used, usually in combination. None of these TKIs, tyrosine kinase inhibitors, actually kill cells. So they put cells into stasis. And so you need a second drug, like an immune checkpoint antibody maybe, or another chemotherapeutic, or a combination of kinase inhibitors to really kill the cells, which is what ultimately you need to do in order to get a cure.
You've seen something leave your lab and turn into a real drug that changes patient lives. Does that experience change the way you see yourself as a scientist? Do you tend to focus more on translation when asking questions now?
I mean, I'm really a basic discovery scientist, and I hadn't really ever done anything translational. I mean, what we did led to translational work. And in fact, we didn't patent any of these original discoveries because it really wasn't common for biologists to patent things. And so, in principle, we could have thought, and I guess we did, that maybe if you could target these kinases, that would be the potential treatment, but we didn't have any experience in how to do that, and that depended on companies. There's been much more emphasis now on being translational. Most applications now require you to say something about how what you're discovering or would discover with funding could be translated. So really very little of what has gone on in my lab has been translational. It's been sort of more blue sky discovery type research, hoping to find something new and interesting. And the work on histidine phosphorylation that we've been doing for the last few years really wasn't originally intended to be translational. It was just to try and understand how this new or different type of protein phosphorylation is used in cells. Ultimately, it may lead to something, but there was nothing immediately on the horizon. The two cancers we have focused on, particularly pancreatic cancer, where we did hope that what we were doing might be translated. And our work on secreted proteins mediate crosstalk between different cell types in pancreatic cancer led to the discovery that the leukemia inhibitory factor, or LIF cytokine, could be potentially targeted, and not through our work, but a small company developed an antibody that neutralizes the LIF cytokine, and that has been in clinical trials in pancreatic cancer. And AstraZeneca acquired this antibody a few years ago now, and they've run a phase 2 trial whose results will be revealed shortly, I hope. And then the other cancer is bladder cancer, where we had this recent work on the PIN1 proline isomerase protein, which we had discovered, or postdoc in the lab had discovered in 1996. And it's been of interest as a potential cancer target for many years. The PIN1 protein in the cancer cells is important for them to be able to make cholesterol, which they need in order to proliferate and survive. And so targeting PIN1, potentially in combination with another drug that also blocks cholesterol synthesis, a statin, is a potential therapeutic avenue there.
You've been able to see quite a few projects develop over long periods of time. Does seeing that evolution of scientific understanding make you research or ask questions differently?
Well, I run the lab by allowing postdocs to pick their own projects. They own the project and they can take it away with them when they leave the lab. And so in the case of PIN1, the postdoc who discovered it, not looking for it specifically, but through studies on another protein kinase that he had worked on as a graduate student, so sometimes postdocs bring projects from their graduate studies. He stumbled on PIN1, showed it was important in cell proliferation, and then he went off to Harvard to be an assistant professor, and he took the project with him. So basically, we didn't do very much more on PIN1 until Shui Wang came to the lab and said she wanted to work on PIN1. And I said, that's great. We haven't worked on it for a while. And she said, I want to work on it in bladder cancer, and we'd never worked on bladder cancer. And so really, the credit goes to her for having decided this was going to be an important target protein in bladder cancer. And she really spearheaded the work on this project by herself.
What made you decide to run your lab that way?
I was sort of brought up in science. That Asher Corner, my thesis advisor, he had 9 students in the lab. He basically said, find something interesting to do. So I pretty much did my graduate project independently. And so having had that experience, I felt, that was a good way to get the best out of people, was to let them find something that they were really passionate about, and then they would put more effort into it, and they could take it with them if they wanted to go to an academic position.
Related to that as well, do you have any other specific ways that you like to establish culture in your lab?
Well, as I say, I encourage people to find their own projects, and they can either pick up something that was left behind by another postdoc or person, or they can come up with something totally novel. People come to the lab because they're interested in the general area of phosphorylation or translational modification, ubiquitination, simulation. And so there's a natural sort of affinity between their interests. And so very often the collaborations arise within the lab that really sort of strengthens a project. And while they may have their own individual projects, they collaborate with other people for specific areas in specific, parts of the project.
Do you collaborate with other faculty yourself?
We have collaborated with many faculty. I think I said 45 faculty members and I have had our names on the same paper, as it were. It's been, a great strength, I think, of both the Institute and my own work.
Facilitating close relationships and collaboration was a huge goal in the founding and design of Salk. Do you think in practice that the Institute accomplishes that?
Absolutely, yes. No, I think we encourage it. When we recruit new people, particularly junior level people, one of the things we think about is will they be collaborative? Will they interact? How well will they interact with other people? Will they take advantage of the fact that we have all this expertise here that would allow them to move and branch out in a new direction? Yeah. In fact, I often tell people as they start out that, they've been recruited to work on what they have been doing as a postdoc. I expect them within a few years to be working on something totally different because of the environment and influence of other people here.
Is there anything specific you look for when interviewing new faculty?
I think the most telling thing is the chalk talk. They always give very professional interview seminars, right? On the work they've been doing. But it's when they have to sit in a room with 20 or 30 other faculty members, grilling them about, what their plans, what they're proposing to do, how well they cope with that environment, and how quickly they think on their feet. I think that's probably the most important. And particularly if they're really good, they will say spontaneously, I think I can collaborate with X or Y on this particular aspect of my project. So they've done their work. I think that's always a good sign.
I'm so curious, culturally, what things do you think have changed the most and the least at Salk since you started 50 years ago?
When I started, it was really a small institution. There were only a couple of hundred people here. We were all, with one exception, in the North building. And so there was a lot more interaction. I think now people are a little more siloed between the North and the South building and the East building. We don't spontaneously see them in the way that we used to. I mean, there are, ways through which people interact. Faculty of the Month talk is one way that you can find out what your colleagues are doing. Faculty lunch, other sort of smaller meetings is a way that people can interact. You won't collaborate unless you know what someone else is doing.
You've won many awards.
All for the same thing.
I'm curious, though, has there been a milestone or award or a thing that happened in your career that anyone else would probably think is silly, but you found particularly meaningful or special?
If you look at my office door, you'll see the occasion when I was mock knighted by Jamie Simon, dressed up as Queen Elizabeth I. And the sword is still hanging over my door that he made for this occasion. That was pretty surprising. Yeah, other important things in my life, obviously our kids, our two sons. Our son has just got an assistant professorship at UCLA. So I'm very proud of him. And so my family has been really important in supporting me. That's not a single occasion, I must admit, but that's perhaps the most unusual one was the surfing award that UCSD gives out. I forget what they call it. It's the Queen Maha Surfing Award. So it's just a picture with a surfboard on it. That was pretty unusual.
Something else I've heard that is quite meaningful to you is your t-shirt collection. That's your real legacy.
Yes, I have a huge collection. It's something close on 400 now, which my wife despairs of because they're taking up so much shelf space in our closet, or closets, actually. I think it probably started with River Trip T-shirts, actually. I have River Trip T-shirts going back to the 70s. First meeting T-shirts would have been in the 80s. A lot of them designed by Jamie Simon. The poster on the wall outside my office on the left-hand side is all of the cartoons he did for abstract book covers for the Oncogene meeting that I started with George Vanderwood in 1985. So, and then he's done T-shirts for all the Salk meetings that I've organized. But T-shirts have gone out of fashion. People really don't. I mean, meetings don't give out T-shirts anymore. Just sort of sad. Yeah. But anyway, that's the way it is.
Do you wear a different t-shirt every day?
I have favorites.
Don't think you could have gotten away with t-shirts every day back in Cambridge.
Absolutely not. I would never be. I mean, I didn't wear ties, but I often had a kerchief when I was a student. Things have got less formal there anyway. I mean, it used to be you had to wear a tie to dinner in hall when you were an undergraduate, and your gown. So when I came back, I was, as I said, a fellow of Christ's College. Formal occasion, you eat at high table, which is on a platform 10 inches above the rest of the hall, and the undergraduates eat on tables that are down the length of the hall, and the high table is across the top.
Just like Hogwarts.
Just like Hogwarts, exactly like Hogwarts, which was shot in Oxford, as you know. And so the fellows come in wearing gowns. You have to wear a gown as well. And so when I came back from California, I'd grown my beard, obviously, and I looked definitely didn't look like a fellow. And I had made some macrame headbands to keep my hair under control. And so I would go and I would go and dine in hall wearing a macrame headband. They didn't say anything, but I did it. So I didn't wear t-shirts to hall.
I feel like that is a perfect way to summarize your style and your attitude and your intellect all at once. So I'll end there. Thank you so much, Tony.
Thank you.
Tony Hunter is truly a force whose impact goes beyond the translation of his findings. By supporting the curiosity of fledgling scientists as they come through his lab, his impact expands exponentially. As he mentors, his mentees become mentors, and on, he shapes generations of cancer biologists. After 50 remarkable years of discovery and countless awards and honors, Tony still enters the room in a graphic tee, flip-flops, and iconic white beard, ready to talk science and recollect memories from inside and outside the lab. Thankfully, he's still making more memories in his well-loved Salk office, where he sits surrounded by a lifetime of books and papers. There, amongst his incredible archive, he stays curious and engaged, seeing discoveries through yesterday, today, and tomorrow. He's already changed the field of cancer biology enormously. Who's to say what he'll find next?
Beyond Lab Walls is a production of the Salk Office of Communications. To hear the latest science stories coming out of Salk, subscribe to our podcast and visit salk.edu to join our new exclusive media channel, Salk Streaming. There, you'll find interviews with our scientists, videos on our recent studies, and public lectures by our world-renowned professors. You can also explore our award-winning magazine, Inside Salk, and join our monthly newsletter to stay up to date on the world within these walls.